Hi everyone, in this lecture I am going to discuss about thin layer chromatography. Thin layer chromatography is a widely used analytical technique for separating non-volatile mixtures. This method is essential in both chemistry and biology as it helps the identification of compounds and analysis of various components within a mixture. This thin layer chromatography is called as thin layer because it consists of a glass plate where a thin layer of stationary phase is coated onto it, and with the help of a mobile phase we can separate different mixtures of non-volatile compounds. The principles of thin layer chromatography is separation based on polarity. So we can separate the mixtures into different individual components based on their polarity, because the polarity will vary for each and every component, and with the help of that we can separate using a suitable solvent system. And the stationary phase, we can use the mostly used stationary phase is silica gel, and other than that we can use alumina fillers, etc. And the mobile phase consists of various solvent mixtures, and the components travel at different rates. As I said, based on the polarity, the components will be separated at a different rate. Whenever the sample is more soluble in the solvent system, it will be eluted at a larger speed to a greater distance in that solvent system. So it will travel faster. But if the solvent is not suitable for the sample, then it will travel at a slower rate. And then finally the visualization is done using UV light in some cases, because UV light is a universal identification method. But other than that we can use another components, chemicals can be used to identify the presence of isolated components. Then TLC procedure. The TLC procedure involves preparation of TLC plate. First we have to prepare our TLC plate. There are some ready-made plates also available, but for laboratory purposes we have to prepare a TLC plate and then apply sample spots. We have to prepare our sample as a solution and apply them as a spot, and then place it in development chamber. And after taking a development chamber, we have to add a mobile phase, and we have to keep our TLC plate inside that, and finally we have to visualize the results. Let's see the procedure in detail. The detailed procedure involve preparation of TLC plates. TLC plates can be prepared by using stationary phase material that is suitable for our sample. If we can use different types of stationary phase materials, and with the help of the individual methods we can prepare our TLC plate. There are different methods available, like pouring method, dipping method, and spreading methods. So with the help of those methods we can prepare our TLC plate, and we have to dry them at room temperature, and then finally activation is done using hot air oven. And after preparing the plate we have to mark the baseline to apply the samples, because we have to know where we have applied our sample. So in at least 1 centimeter or 2 centimeter distance we have to draw a baseline, and we have to give a spot using a pencil, because the ink of pen will also flow along with the solvent. So baseline is usually done with pencil, and the sample mark should also be identified. It should be marked using pencil. Then with the help of capillary tubes we have to keep our sample spots, and finally we have to prepare a mobile phase solvent, the solvent that can be suitable for separating a mixture of components can be used as a mobile phase solvent. And with the help of either development chamber or in a beaker we have to prepare our mobile phase solvent, and we have to allow the mobile phase solvent to be at a room temperature for some amount of time to attain saturation, and after that we have to keep our TLC plate inside that. And after development we have to take our TLC plate out, and we have to spray some reagents if we are going to use specific methods. But if we are going to use some non-specific methods, we can use UV radiation. We can identify the spots in UV chamber, or with the help of some other reagents like sulfuric acid reagent and iodine sublime iodine also we can use. Applications of thin layer chromatography. With the help of thin layer chromatography, we can analyze the pigments present in plants, and we can also monitor the drug development processes, and for food analysis for contaminants. The advantages of TLC include simple and quick technique. It is a simple one. We have to prepare the TLC plate, and we have to prepare the sample solution, and with the help of a universal visualization method we can identify the presence of the components. So it is simple and quick technique, and the minimal equipment is required. There is no sophisticated equipments required for performing TLC. We just need to have a development chamber, a glass slide, and that's all. Then cost effective for labs. Then high resolution of separated components. Components can be easily separated, easy to operate and interpret. In conclusion, it is a valuable analytical tool, and various scientific fields also they are using TLC methods, and it also enhances the applications. Thank you.